Good morning, everybody. I'd like to thank Timo and Alan and the organizers for inviting me and giving me this opportunity to go over some of the recent results we've had looking at connectivity of the cerebral cortex. In the last few years, it's become increasingly fashionable to think of cortical organization on large scales in terms of its spatial embedding. It's not always clear to everybody exactly what one means by that, but in simple terms, it's not thinking in terms of binary networks to understand in a predictive fashion exactly what is the organizational principles of the cortex. So this notion of spatial embedding requires that we have an understanding of the weight and the distance of connections to understand the organizational principles that lie behind the cerebral cortex. And in line with that, we collaborated with a number of very gifted people to develop these large-scale models. And we were very, very confident that we had put our finger on a defining principle that allowed us to have a better understanding of cortical organization. And then it dawned on us that we would be making a terrible mistake. We had this idea that the strength and location in space of connections in the cortex is a determining feature, which gives you this predictive power to observe numerous features, but we were treating cortical areas as single entities. And we know that's not true. We know that areas have an extension. They have a physical extension. And we know that the properties across the extension changes in a regular fashion. We know that the cortex is organized according to gradients. And these cortical areas, which we're so fond of, are actually abrupt changes in the steepness of those gradients. So we were making a cardinal mistake in treating cortical areas as these single entities. And we designed the networks based on this by forming beautiful figures that we put in various journals, where cortical areas are connected by their barycenters. A barycenter is a mathematical center of something, in this occasion, the cortical area. But of course, we know that cortical areas don't communicate by barycenters. So we had this ludicrous situation where we were preaching the gospel of spatial embedding with the whole notion of a cortical area as a point, which it's not. Every electrophysiologist which has looked at every cortical area imaginable has noted variation of receptive field properties in regular fashions. And this is equally true for the visual cortex. The title of the presentation I'd like to make today is How Retinal Eccentricity Shapes the Macaque Cortical Connectome. I sort of put macaque there because the results I'm going to be sharing with you are looking at the macaque cortex. And the macaque cortex is dominated by vision in a way that perhaps the human cortex is not. So the results I will show is that when we forbear this thinking of area V1 as a point and start taking into consideration its spatial extent, we're going to find connectivity changes across the extent of visual cortex, which indeed serves to shape the connectome of the macaque cortex. It might do so in a way more that might not be exactly what you would find in a human if you could do the same sort of thing. To be polite, I'm going to go through the procedure of showing you some of the data, but this is anatomical data, and it makes for very, very busy slides, and I'm not sure that we would really have the time. I thought the presentation was going to be 15 minutes, and I was so sure that 15 minutes would not allow us to see this anatomical data. So I'll go for it pretty quickly, but I'm going to just go over a little bit more about this weight-distance connectivity and how it predicts numerous features of the cortex and why we call this spatial embedding. I will go through that to some extent. I've mentioned already that we're going to now take into account the spatial extent of cortical areas. And if I had the time, and I won't, I would really like to talk to you about cortical hierarchy, because I think everybody in this room is very functional orientated. And the anatomist's handle into function, of course, is looking at hierarchy. It's the cortical hierarchy now, which from the work of David Van Essen, but then very much more recently in the last 20 years, where people have been asking, what is it that's going from one hierarchical level to the next? And I think that this anatomy has had a lot to say about that regularity. So I won't have time, but I would like to. And then finally, the claustrum projects everywhere. It is actually the strongest connection, let's call it subcortical. Herculana Husel, you'll forgive me for that. But it's not a cortical area, at least. It's paleo, but it's not a cortical area. It's the strongest subcortical projection to any cortical area. And it disobeys this spatial embedding principle, this weight distance. It disobeys that. Its projection is equally strong to nearby cortical areas and cortical areas which are right at the back of the room. So the claustrum would normally be a way of challenging the principles that I would like to show. So the techniques we've been using are extremely old-fashioned. They're tracing connections with retrograde tracers. These are chemical tracers, and they're extremely robust, and they have the advantage of working. And they have the extraordinary advantage over diffusion tractography in that they give meaningful results. I hope nobody's upset by that. Meaningful results in the sense that if you look at area V1, which is the primary visual area of the primate cortex, and you do a tracer injection in 2011, and people have been doing this for donkey's years, you can still find connections which have not previously been found. And I think that means something. This is really the sociology of large-scale models over the last 20, 30 years. This is the Feldman-Van Essen model, which has been so dominant. It's a collated connectivity database, which David and his student used to propose the famous Van Essen model of hierarchy. And this is the model that we produced a number of years later in collaboration with David and others. And we have a density here of 67%. The density that David was using was something around 35%. The density of the graph is the percentage of connections that can exist that actually do exist. So with a density of 67%, it means 2/3 of the possible connections are actually there. Now, the density of the connection of the graph is a cardinal feature that determines numerous features that we're not going to go into at any length. But when we made this publication, this is a review in Science in 2013, the dominant model which was being used at the time was a small world model, where people were claiming that small world networks were very useful for understanding the cortex. They're very useful for understanding social networks, because the density of the social community of humanity is something like 0.000 something. In a density of 67%, to say that it's a small world is actually rather meaningless. It's not rather meaningless, it really is meaningless. And this shows you a number of studies which had used the same database that David had, but did strange things to it. And eventually, it ended up with a paper by Motta and Singe in 2010, published in PNS, which had this flag of the small world network, and they managed to get a density of about 6%. So you could argue maybe a small world network is interesting at that density. But actually, the data was meaningless. So you kind of done the circle as you wish. The mathematical regularity, which is really useful for designing these large-scale networks, which shows this spatial embedding principle described here. And in a nutshell, you have this log-normal distribution, and you find this log-normal distribution at the mesoscale level. This is strength of connections. but you also find it at the single cell level. You have an exponential decrease in strength of connection with distance. This is perhaps the cardinal feature. And you have this geometry feature, reflecting feature, which is the Gaussian distribution of distances. And for Zoltan Torokai and his colleagues with whom we collaborated with, these features strongly suggested the spatial embedding that we were able later to develop. So the way to do this in a very simple fashion is to look at motifs. This is what I had on the previous slide. You've all got lots of neurons, so you captured that. And when you do these, if you look at these motifs in the real brain, you find that the models that we've been building predict exactly the same motif distribution. So this is a flat map from David Van Eschen, and it illustrates, I think, this problem of let's stop using cortical areas as points. Let's think of the extension of the cortical area. This flat map represents V1 here and V2 here. V1 is in this peculiar shape, but this is all folded, and of course it lies contingently to V2. Let's not get too worried about that, but the point I want to make is that this is the foveal representation, and the foveal representation, as Leslie Ungerleider remarked so many years ago, is in front of what we like to call the ventral stream. And the peripheral representation of V2 and V1 is in front of the parietal cortex, where we have what is known as the dorsal stream, which is located like this. And so Leslie Ungerleider was able to show that if you looked at the connectivity of the dorsal and ventral stream cortical areas, you found that they were preferentially related to, connected to some extent, to the foveal representation and the lower periphery. The anatomy that I probably won't get round to actually sharing with you is going to demonstrate that this, we're now going to be looking at the feedback projection. So we're doing tracer injections in V1 and V2, and we're looking at the feedback coming into these areas. So remember, in this computational age, feedback is now thought of as relaying descending predictions, expectations. And the feed-forward pathways are meant to be, are considered to be the unresolved issue from the lower level, i.e. the prediction error in the predictive coding. So in that context, the point we're going to be making is that this foveal representation here of V1 and V2 is in a very, very, very different environment from the far peripheral representation. And so you would predict that the projections back to these two different regions should be very different, and they are. And that's the essence of what I'm going to present. And there's two regions of areas V1 and V2. You have a dorsal region, which represents the lower visual field, and you have a ventral region, which represents the upper visual field. And at the end of my presentation, I want to come back to an extremely interesting publication in 1991 by Fred Previck, who argued in favor of an ecological theory of vision. It's an extraordinarily interesting paper. And what he's saying is that basically you can think of two aspects, and you know how the human brain loves this, 2, 1 and 2, dorsal and ventral. But you can also think of peripersonal space and extrapersonal space. And he argued very convincingly that the data in visual science up to 1990 argued that the upper visual field and the fovea would be dominated by processes which are to do with extrapersonal space, far vision. And the lower visual field would be dominated by peripersonal space. So if you're a mouse, it's the ground rushing behind you, rushing underneath you. But if you're a monkey or a human being mending a watch, it would be what your hand is doing. It's threading that needle, that thread through a needle. And so he argued that this would be what you would expect to be the case. The anatomy that I'm probably not going to show you, but you can believe me, because why not, is that the actually
Take the 5 minutes.
the upper visual field and the fovea share a very similar connectivity, and the lower visual field is standing right out of that. But because I'm in a meeting dealing with the frontal cortex and the prefrontal cortex, I'm going to also be able to show you that if we look at what this connectivity implicates in terms of the connectome across the cortex, it has profound effects on far-flung regions. So these are the sort of networks that we were building with, these points, and we're not going to come back to that. This is just to give you a brief view of what is the actual extent of what we're calling the dorsal stream cortex, because I'll be talking a lot about this, and the ventral stream, and these are the injection sites. This phenotypic map has been done by David Van Essen and Yuji Ho in the team, and is now being revised. The map is based on recordings that David was making back in the last century, but we need to update this, and we're in the process of doing this with Wim van Duffel, I think is here today. But we haven't had time to remap all the data, so there will be some little changes as we go along. So here's the anatomy. This is this is area V1, central representation. We've made a tracer injection here. This is that dorsal stream in pale blue. This is the ventral stream here in pale yellow. And you can see that there's a dominant connectivity coming back from, these are the projections from the ventral stream to the injection site. These are the feedback connections. And when we go to the far periphery here, you can see that here, yes, there's an overrepresentation of neurons in the dorsal stream. So that premise that the proximity of the dorsal and ventral stream would dictate the connectivity of the upper and lower visual cortex, the lower and upper visual field representations in the cortex, is substantiated. If we do an analytical representation of this, you can see that there's more of this. So this is a very busy slide. This is the representation of V1, which we're taking as sort of the basic kind of ground truth, if you were. This is what we've been dealing with over the years. And this is the representation of the connectivity that is resulting from a foveal injection. And if you now come to the other injections at other eccentricities, so the far periphery upper field, the far periphery lower field, all in area V1, the red outline gives you the connections which are not represented in the foveal projection. So these are all connections which are non-existent in the red line. And here there's color-coded, so that the yellow means upper connections only going from upper to upper visual field, and the blue uniquely to the lower visual field. And you can see that this is a very busy slide. There's many connections which you're not observing. You're not observing in the foveal representation. And this is even more true. If you look now, this is exactly the same way of looking at things. This is area V2. This is the extent of the non-foveal projections to the far periphery upper field, far periphery lower field, and these are the color-coded, and you can see that there's a large number of areas which are strongly connected with upper and lower visual field. And if we quantify this across the representations, you see that there's a dorsal stream domination in all cases in the V2 and V1 lower field, but all of the other instances, the V1 paracentra, the V2 center, the V1 center, the V2 paracenter, and the V1 far periphery upper field, and so we haven't got a complete coverage, is doing exactly what Fred Privik's paper predicted. It's showing that there's an exception for the lower visual field. So we began to look at this, and this is perhaps the most exciting part of the presentation, and one in which I feel least armed to really go into it because this has only just been done. We're doing this work with Slavoj Horvat and Ravas Erskes and Zoltan Torikaj. So we've done a Voronoi diagram, a Voronoi partition, and this enables to look at the shortest distance trees which are rooted either in V1 upper or lower field. And the outgoing would be coming out of that, and the ingoing is coming into that being V1. Now, to make a tree like this, of course, we have to include injections which have been made throughout the whole of the cortex, which is what we've done, and I'm not going to present that data because we haven't really properly analyzed it, but we have some results that we can share with you. And these shortest distance trees, the shortest distance is the strongest connection that can connect any two areas across the graph. We call it the shortest distance, but it's the strongest connection. So this is the distribution, and it's color-coded. So I've lost the mouse. Can anybody see it? I really don't have enough neurons for this, huh? So the blue is color-coded. It's the upper visual field. This is the upper visual field. And what the Voronoi distribution is saying is that this orbital frontal cortex is very dominated by this. The green is the central representation, the fovea representation, the representation that we've been looking at systematically until now. So this is the fovea representation, and it's what the anatomy showed you. I mean, I went very quickly, but it's very, very, very restricted compared to the lower visual field and the upper visual field. And the lower visual field and the upper visual field have very, very different domains. Very different domains. These are the shortest connections, and the trees going in and going out are not identical, so you don't find exactly the same pattern. There's nothing peculiar about that. I mean, it seems peculiar at first, but I've looked at this with Slavoj Horvat, and he's convinced me that this is not peculiar. But it's showing you that you have very, very, very different domains. So these are the shortest distances going back into these different regions of V1. And that means they're relaying in a down projection expectations of a different nature. The expectations coming into the upper visual field are not going to be the same as the expectations coming into the lower visual field. And the expectations of the foveal representation are extremely restricted. So it really underlines the fact that you can't afford at all to think of V1 fovea as representative or proxy, is that another word, of connectivity or what have you. Anything based on that is going to be totally inadequate. So let's see if this works. So what Horvat has done here, Slavoj Horvat, a very gifted guy who's at the moment in Max Planck in Dresden, he superimposed on this Voronoi partition the actual trees, the shortest distance trees. So that means that when you have a long distance connection like that, that means that projection actually exists, and it's the shortest way, because if you do individual hops, you're going to be losing efficiency, if you were. So it's a way of imagining exactly what this differential connectivity in these different streams is going to give you as a consequence. This is just to tell you that we have very robust data showing that the projections back into V1 and V2 are eccentricity dependent. So if you're looking at projections into the fovea, we know this from a long way back. They're not of the same ilk as they are if you're looking at the projections into the periphery. Because actually, what I really wanted to say is the hierarchy is really what the anatomy can tell us about. That's really interesting for the functional people, which is what I'm addressing. And we know there's two flavors in the feedback projection into lower visual cortex. There's a supragranular and an infragranular, and they are not at all the same in the peripheral and central representation. So this is a slide of the credits. There's 2 teams. There's one in Lyon, and there's one in Shanghai. Ming-Li and Yuji Ho, who's here today, are the lead authors on the publication we've been talking about. There's been many, many very gifted people, Anna Gomez, Loïc Magrou, Julien Vizoli, who's here today, who've made, and Ken Knobloch, and Colette DeHay, who's been very supportive, who've made very good contributions to this work. In Shanghai, the team there is headed by Ziming Shen, and Muming Pu has been strongly supportive of this work. The ability to do what I've been showing you has been made largely possible because of this team of people, Takula Hayashi, who's in Japan, Matt Glasser and David Van Essen at WashU. And the network analysis has been done by, to a large part by Shobot Horvat, but Maria Escaro Varhavas has been extremely helpful with that, and Zoltan Torikai. We were able to do an imputation of beautiful quality imputation, so that the networks I've been showing you are of a fully connected graph. Thank you for your attention.